Okay, hello. Good afternoon or good morning. We would like to welcome you today at this Alba Fence webinar. It's a joint webinar, it's a collaboration about understanding sex-based differences in brain research. So as you probably know, most psychiatric and neurological disorders manifest differently between men and women. But historically, research has often neglected these sex differences, and sometimes also in understanding the disorders or treatment effects. Fortunately, nowadays, there are many guidelines from the National Institute of Health and the European Commission and others that need to include sex as a biological variable in research. So today we'll talk about preclinical and clinical research and how we can incorporate SABV, sex as a biological variable. And with that, I would like to introduce our first speaker, which is a distinguished researcher and a dear friend. And this is Dr. Georgia Hodges, that Georgia received her bachelor's degree in drama from Bard College, and she worked as an actor in New York City. Then she went back to school and she obtained her degree in psychology at the Hunter College, and then a post-graduate degree, like a PhD, at Rutgers University in the Behavioral System Neuroscience Division of the Psychology Program with Professor Tracy Source. Of course, there she worked on sex differences. She will tell us some of her research later today. Then after her PhD, she went on to have a postdoc in pharmacology in the lab of Dr. Elaki at the University of Pennsylvania. And then in 2010, at the lab of Dr. Scott Russo at Mount Sinai. And in 2013, she received the NARSAD Young Investigator Award. And in 2015, she was promoted as an assistant professor at Mount Sinai. And now she's a faculty member at the Virginia Tech since 2016 as an assistant professor. And she will talk to us about sex differences in preclinical research mostly and how to incorporate some. So
thank you so much for that introduction, Christina. I'm going to go ahead and share my screen. All right. Everyone can see it okay? Great. So I'm going to be talking today about sex as a biological variable. And I really want to go over some of the history of why we're looking at sex as a biological variable, particularly in preclinical research, and where we need to go from here. So, sorry, just moving this around a little. I wanted to start with why women were excluded from drug trials. And so this really started in the 1970s due to a drug called thalidomide that was given mostly to pregnant women in Canada and Europe. It was never approved in the US. But because of the birth defects that were triggered by thalidomide, it was thought that women should not be a part of clinical trials, particularly phase 1 and early phase 2 drug trials. And you know, this was obviously protective for the health of the children, but there was also this concern that even that women who weren't pregnant would eventually become pregnant, and something that they had taken in these drug trials would affect that pregnancy or offspring. And as a result, women's health got sorely neglected for both clinical trials and preclinical trials. So in the 1990s, this was changed, that the National Institute of Health ends up establishing the Office of Research into Women's Health to push for adherence. There's a Women's Health Initiative in 1991, and then in 1993, Congress writes this NIH inclusion policy into federal law, which requires female subjects to be in all NIH-funded analysis. And in the 94, there are revised guidelines that sort of push or comply anyone who's receiving a grant or working through a cooperative agreement to include human female subjects. And so clinical science starts to move forward, but there's no such mandate for preclinical research. So the idea was, now we're including women in these studies, everything is okay. However, in about 2001, the US Government Accountability Office issues this report that says that out of the 10 drugs that were recalled in the past few years, 8 of them were recalled solely for their adverse effects in women, right? So these drugs that were designed in male animals and then tested in male and female populations and seem to work in both may have very different effects in women than in men. So why? Well, one of the reasons for this is that little to no preclinical research was done in female subjects. So this is a composite study starting from 1992, looking at subjects in preclinical studies. In orange is female-only subjects, light blue is male-only, and dark blue is both sexes, with gray being not reported. And as you can see, in about 1992, the majority of studies were either not reported, meaning most likely they were in male subjects, or were in solely male subjects. And this starts to change in 2009, but still it's a pretty equal amount. As you can see here, looking at 2009 versus 2019, there's not really that much of a change in male-only subjects. What ends up becoming changed over time, particularly with the mandate in 2016 from the NIH that we need to use female subjects in preclinical studies, is that now more of the sex is reported, right? So we get this decrease in 2017 in the amount not reported. However, the number of male subjects or both is increased, particularly by 2019. And if anything, female-only subjects or studies with female-only subjects declines from 1992 to 2019. And that's really problematic because there's a lot of conditions that only affect women, right? And we're not necessarily doing these studies now into things like the effects of lifelong hormonal contraception on the brain or menopause, because we're not doing studies that are only focused on female subjects. And in fact, one of the problems has been that in using sex as a biological variable, people get comments back on papers and grants saying, well, you're not looking in males for female-only conditions. So I think moving forward, one of the things that we need to think about is having real women's health initiatives in addition to using sex as a biological variable. So why we need to use sex as a biological variable is really summed up nicely in this chart of drugs that act on CRF as CRF inhibitors. So all of the preclinical modeling was done in male animals in these studies, and these worked excellently in the preclinical studies. But when they were brought to clinical trials, they did not work. And part of that may be because in clinical development, we're looking at a population generally of people who have mood disorders, and the majority of those people are women. And so when this was tested in groups of clinical studies that were either half male, half female, or majority female, as I think you can see from the X's, these drugs didn't work. In fact, the only trial where this worked was a trial where they looked exclusively at human male participants. And so it starts to get into this idea that there are sex-specific drugs, right, that we may need to individualize drug treatment. And certainly we need to individualize how we're doing our preclinical and clinical studies, right? That if we work only in male animals, and then we try and translate this to human subjects where it's majority female subjects, we are not going to find an effective drug. Whereas if now we start with preclinical studies that are male and female, we can either translate that into clinical studies where we look at combined male and female populations, or we look separately at males and females in those clinical populations. And now we may be better at translating what we're finding preclinically into clinical studies. So one of the main reasons that females weren't looked at was there's this concept that there was greater variability in females due to their hormonal cycling. Now, this, in fact, neglects the fact that males also cycle hormonally, albeit more diurnally. But in the 2000s, Jill Becker, Brian Pendergast, and Liang all came together and did this massive study across meta-analysis looking at variability across all these different aspects of neuroscience. So behavior, electrophysiology, histology, neurochemistry. And what they found was that there weren't any differences in variability between males and females across all these different traits. Where they did see significant differences were for non-brain measures. And particularly, this ended up being for things like cardiology and blood and organs, rather than even body weight and endocrinology measures. Additionally, there's this question of the estrus cycle. Right. So this has been where this variability is supposed to come from. And so they looked again at trait variability across studies and found no significant effect of the estrus cycle or sex differences in trait variability. Now, even when the cycle was taken into consideration. However, this does not mean we just ignore the estrus cycle. I think one of the best suggestions that we can give to people who are interested in studying sex as a biological variable is to look at the variability in your subjects. So if you're within subjects, variability is greater than your between subjects variability. So if within females, the variability is greater than between males and females, then I think you need to start by looking at the estrus cycle. However, there can be plenty of other things that account for within subjects variability, including housing conditions, bedding, and food. But as a rule of thumb, where I start when I start my studies is to look at that variability within subjects to see if I need to now take account for the estrus cycle. And I'll track the cycle, but not necessarily on a day-to-day basis or consider it in my analysis. So how do we include sex as biological variable? So there's this wonderful paper that came out in Journal of Neuroscience Methods with this link to videos about how to design studies and things that we should take into consideration as we design our preclinical studies. The link is also in the paper as well as on this slide. And in talking about this, where we start is really this default for including sex as a biological variable, which is to do experiments with males and females, right? To have a control group and an experimental group where we have equal numbers of males and females. And we can look at this, in control and experimental groups, but also we can do this in studies that have historical data in males only. So we can look at studies that were done only in males and then come back to them and repeat these studies in females with a subgroup of males. And so this allows us to determine if we're really getting validity from what's happening in the previous studies in males and start to look at whether this is different in females. We can look at different aspects of sex as a biological variable. So are males and females behaving similarly, or are they behaving different? We can look at this at a population level. So the percent of mice affected by our treatment and intervention within male and female populations. I think a really exciting area has been convergent differences, where we actually see the same behavior in males and females, but it's caused by different mechanisms. And so I'm going to discuss a study in a little bit that actually is a convergent difference study, where it's really important to think about mechanism even when behaviors are the same. We can look at qualitative differences. Is behavior sex specific? Are animals engaging in lordosis versus mounting? Are they engaging in aggression or maternal behavior? And then finally, we can look at quantitative differences. Are we seeing more in one sex than another? So we see the same type of effect, maybe an increase in function of the adrenal glands or basal corticosterone levels, but to a higher degree in one sex versus another. So I want to switch gears a little and talk about some of the research that I've been doing over the past 20 years or such, looking at sex as a biological variable and what I've learned from it. So first and foremost, I would say that there are sex differences in the behavioral response to stress. Currently, the stress that I've been working with is a variable stress. So this involves a combination of foot shock, tail suspension, and restraint over different days. And you can do this for 6 days or for 28 days. So 6 days is subchronic, it's less than 2 weeks, which is generally what's thought of as chronic stress, or you can do this for 21 to 28 days. And we look at a variety or a battery of tests, so things like the splash test, where we're looking at grooming behavior after being sprayed with a sticky solution. And so we look at this because in both animals and humans, when they are stressed, generally self-grooming habits or self-care goes out the window. We look at a test called novelty suppressed feeding, and where exposure to an anxiety-provoking new environment can change an animal's feeding behavior, even when they've been food deprived overnight. And we look at tests like the forced swim test, which is a test of active versus passive coping. And what I really like about this particular stress and behavioral test battery is that we can create composite stress susceptibility scores across these tests to see how animals are behaving, similar to what we see when we do human studies. And if we look at control males and females, we don't see a difference generally in this behavior. However, after 6 days of stress, we see increased stress susceptibility in females, which we do not see in males. So now we can look at what's the mechanism of this earlier sex difference in stress susceptibility in females. What's making these females more vulnerable to stress at this time? What's making males resilient? And we can look at 28 days of stress, where now we're seeing stress effects in both, and ask, is the mechanism the same? So first and foremost, this is work done by a number of different people across labs. There has been definition of the circuits that are involved in stress susceptibility and resilience. And there's been identification that different circuits or strength of signaling between circuits changes in males and females. Particularly, most recent studies have identified that signaling of the medial prefrontal cortex and the hippocampus, ventral hippocampus, to the nucleus accumbens, which is this area of emotional processing that we know is involved in reward behavior, is stronger in females compared to males. So this increased signaling between these brain areas or increased excitability seems to be a factor in stress susceptibility between males and females at the 6-day time point. Additionally, early studies I did in the Russo lab in about 2010 were looking at sex differences using the stress paradigm after 6 days of stress in transcription in the nucleus accumbens. And what I think you can appreciate here is that the transcriptional signatures or genes that are changed by stress in males and females barely overlap. And particularly, actually, we're seeing more genes downregulated in males. This is the time point where they are stress resilient, suggesting there's this active coping mechanism in males. And this led to a study in humans who had major depressive disorder from postmortem tissue that we did in collaboration with the Nestler lab, where we found that across brain regions, when we compared males with major depression to healthy controls and females with major depression to healthy controls, we see barely any overlap in the genes that are undergoing transcription across brain areas. And this is really important because if we're going to develop treatments for these disorders, and we're targeting a single mechanism in the brain that we're learning from a preclinical study, it's not necessarily going to be the same mechanism in males and females, even when their behavior is similar. And then moving into an analysis of what we saw in the nucleus accumbens, both in humans and in mice, where we really see these differences is in immune function, that we see increased upregulated immune function in females compared to males, at least in the nucleus accumbens, and a downregulation of things that provide support from immune responses in females compared to males. So moving into trying to understand what this might be in the brain of males and females in my own lab, we've looked at epigenetic regulation. So this is a study where we looked at the sequencing data, and then we also knocked out this transcriptional modulator, so DNA methyltransferase 3A that leads to DNA methylation. And I think one of the things that you can see here is that a lot of the top pathways that were altered by stress in the females are immune signaling pathways, and that if we knock out DNMT3A in females, we make them more transcriptionally similar to males. And we were able to block the effects of stress on their behavior doing this. In my own lab, I've started to look at immune cells and trying to understand which immune cells may be involved in these effects. So this is from a recently published study where we looked at how 6 days of variable stress or 28 days of variable stress shift the morphology of microglia. And I hope what you can see here in red is that we're only seeing amoeboid cells in the nucleus accumbens of females after 6 days of stress or 28 days of stress, that we're not seeing these changes at all in males. And this is also demonstrated here by looking at average area and branch length, so that we're getting this decrease in branch length of microglia, meaning they're pulling in their processes. Our most recent work has looked at microglia counts, which are the red cells here. So in the stained green are astrocytes, and red are microglia, and blue are the nuclei of cells, so neurons and other cell types. And as you can see that there's fewer microglia in the nucleus accumbens of females after they go through 6 days stress, but there's also really this dramatic decrease in astrocyte markers. And so this is a particular marker called GFAP. And what we think is happening is that both astrocytes and microglia are pulling in their processes and moving away from each other. And so part of what I've been starting to study is how these non-neuronal cells communicate with each other and how these may be involved in sex differences in the stress effect. So I just want to close with a quick mention of where do we go from here? So what I've talked about with you guys today is how in 1993, we started adding women to clinical trials. And in 2016, we moved forward and started adding female subjects to preclinical trials to try and get better drug development. However, most of the studies we've done have sort of focused on if there are sex differences. And as we have a greater understanding now that there are sex differences across a variety of different types of research, I think we need to start questioning why there are these sex differences. How can we move forward? Can we look at things like that are female specific, so like hormonal birth control and how it affects women across the lifespan? How puberty may be different in males and females, and how this also can affect individuals across the lifespan, as well as really designing and analyzing our data and stratifying by sex. And finally, we really need to start thinking about including transsexual and intersex individuals into our clinical studies and or modeling this in our animal studies so that we can address everyone's health, not just males or females or what we've traditionally thought of as males and females. In closing, I just want to thank the members of my lab, our funding sources, and particularly I wanted to mention my former postdoc, Jennifer Rainville, who's now an assistant professor, my former graduate student, Masha Seglovia, who is now a postdoc at University of Pittsburgh, my current graduate student, Dawson Kropp, and undergraduates, Alyssa Johnson and Sachi Watson, who have helped with these studies. I'll be happy to answer questions. Thank you.
Thank you so much, Georgia. That was a great presentation, and if you do have any questions, we would invite you to just pop them in the Q&A box that you should be able to see. So it's my pleasure to welcome our second speaker in this Alba Fens jointly organised webinar. So Dr. Laura Castro is a neuroscientist and serves as the Scientific and Medical Affairs Project Manager for the Women's Brain Project, where she leads the organisation's scientific projects and manages the Global Women's Brain Project multi-stakeholder collaborations. And after completing her MSc in Biotechnology and Enterprise and then a PhD in Neuroscience at the University of Manchester in the UK, she then went over to Sweden, where she worked as a postdoctoral researcher in the Department of Neurogeriatrics and Medicine, studying mitochondrial dysfunction in Alzheimer's disease. Then, with a funded postdoc position, she delved deeper into the exciting world of cell and gene therapy. She's worked in the pharmaceutical sector as well, and in addition to her research roles, she shares her knowledge as a visiting lecturer in the Monterrey Institute of Technology and Higher Education in Mexico. So since joining the Women's Brain Project in 2020, she's been applying her expertise to further the organisation's mission by leading the scientific team to produce peer-reviewed publications as well. So it's my pleasure to welcome you, Laura. Thank you again.
Thank you everyone, and thanks for the attendees for being here. We have more than 100, which is very exciting. I'm going to start sharing my screen. Okay. Please give me a thumbs up if excellent. Okay, so good afternoon, everyone. Yes, indeed, my name is Laura Castro, and for today, I would like, first of all, to thank the organizers to give the Women's Brain Project the opportunity to speak about what we have in our pipeline and the exciting journey that we are experiencing. So these are my disclosures, and I would like to start by saying, who are we? So the Women's Brain Project is a nonprofit association. We were founded in 2017. We are based in Switzerland, but we have a global outreach where we work together with experts in different fields and different stakeholders, including patients, the relatives, and policymakers. And we advocate and we are trying to understand the underlying sex and gender differences in neurodegenerative, but also neuropsychiatric diseases. At this moment, we are transforming into a foundation to operate the first sex and gender precision medicine research institute. And we have global collaborations. We are really active in social media, and we have won different prestigious awards, and I encourage you to go over to our webpage and check out our scientific publications. And basically how we are structured and which are our four different pillars are really building up on preclinical, clinical research. We also partner and co-create to bring pieces regarding regulatory frameworks, but also studying the socioeconomic dimensions and the policy and the advocacy work to rise sex and gender matters. But also we have as well partners that where we are, that they are allowing us to investigate with them and co-create pieces regarding novel technologies and AI. And these four pillars work together to actually consolidate our mission, which is transform into a foundation that will operate the Research Institute for Sex and Gender Precision Medicine. So I want to show you our current model. Today, we are co-creation with different partners for research projects, papers, and European grants. Also, we partnered with pharma companies, and as I said, we have socioeconomic projects that study their implications. We have as well policy and advocacy projects, and we partnered for investigating sex and gender implications in novel technologies. So if you are interested into knowing more about the history and some of our more seminal works, you can obviously go to our website and you can have a look at all our scientific publications. But I will strongly highlight that in these publications, I try to cover all and summarize all our seminal work. So this is, if you don't want to read all our papers, you can just read this one and have a taste about what we are doing in the Women's Brain Project. And as Professor Georgia said, actually we have different sex difference in PK and PDEs regarding distribution, absorption, metabolism, elimination, etc. We already really know, and we have evidence about this, that we have differences in how the efficacy and the effectiveness of some antipsychotic drug works in women compared to men. And we already know that we have adverse drug reactions for more in women compared to men. So this has come with an economic burden, right? But actually, if you go to the literature, you will see that we have excellent pieces of work highlighting the PK and PD, and research actually start to ramp up in the latest years, actually highlighting the sex difference, the innate sex difference, the biological sex difference that we have in distribution, absorption, metabolism, etc. And what is really shocking to me, and this is actually why I do what I do, is that yet we have all this evidence. However, national guidelines and summaries of product characteristics, for example, for antipsychotic prescriptions generally do not have female-specific dosing recommendations. So I think that the change, we are in a really exciting state as this change is starting to be in the policy agenda of some regulators, for example. And I would like to give you a flavor about some of our publications on some of the pipeline that we are doing at the Women's Brain Project. And one of these one, and which is actually related to Professor Rhodes, was that we went into the literature and we checked whether we have evidence regarding sex difference in sacral unruptured intracranial aneurysms. And what we saw is that indeed we have evidence. However, we don't have most of the evidence that we found are regarding the aneurysm growth that is different between male and females. We have different risks, for example, and the risk of multiple aneurysms. However, when we investigate and check and research whether the endogenous estrogen levels are maybe one of the drivers of increasing the risk of developing of UII, we couldn't find a massive scientific evidence. And I think this is really linked to what we have previously said, and this is because it's actually difficult, and basically the question whether we can incorporate, or we should, I would say, incorporate men and females in design of an experiment, it was maybe not in our minds. And I will say that in my perspective as a neuroscientist, is not if we should do it, is that we need to do it. And this will therefore translate in having less noisy data, for example. And when designing experimental hypotheses, I think is a step where we need to really go first into the literature and formulate an hypothesis, and therefore an experimental setup that allows to answer that hypothesis. So also regarding real world evidence, I was into the literature and also linked the monitoring of endogenous estrogen levels. Now I come across with these two papers, and these two papers show that using these wearables, aptamers, sensors, we are capable of gathering data in picomolar levels of estradiol during the fertility cycles of the females. So I was wondering why we cannot gather the same real world evidence data using these wearables and extrapolate them to neurodegenerative diseases such as Alzheimer's disease. And now an idea that I'm just putting into the table, and I think wearables will allow us actually to capture this. In terms of policy, as I mentioned, policy is one of our pillars. We have just published this year one paper highlighting why we are as I say, in a very exciting time period where we need to show that brain health, by prioritizing, by having a global synergetic action to prioritize brain health, we can actually help driving forward the 17 Sustainable Development Goals set out by the United Nations to end poverty and drive economic growth. And we just have good news today that we have another paper that was submitted and today accepted, which actually is the second step of our previous paper. And in here, this health policy paper outlines 6 strategic drivers to expedite and facilitate a comprehensive national delivery strategy to prioritize the global action plan on epilepsy and other neurological disorders, and optimize and put across the whole lifetime brain health as a priority. So at the Women's Brain Project, we are also making an economic case. We have previously investigated a patient journey for Alzheimer's disease. And in this case, we are making the economic case and investigating whether we have sex and gender differences in Alzheimer's disease and multiple sclerosis. In this case, we want to know to what extent prevention interventions can be tailored for the different sexes, but actually which is going to be the economic investment and return of investment. And I think that in my perspective, the message is clear. And I think that brain health matters to all of us. All of us. It's part of us, it's part of our holistic composition as a human being, and we need to prioritize it. So finally, I would like to show you this slide where where I collated just for this webinar, really, as I know that I will have scientists over here, some of the key publications that I believe there are a must to read for actually implementing sex as a biological variable, but actually why brain health matters and why brain health and mental health shouldn't be cataloged as two separate entities, are actually two entities that feed upon their other. And also this QR code will redirect you to our website. And I say finally, but I forgot that we have a video. So I would like to share with you for the first time our video that shows our transformation to a foundation. So just bear with me a second. In the meanwhile, I am presenting this video. Share. Okay, so turn on original sound.
Do you just want to hit the full screen button just so we can see it? It's full.
Did you know that 2/3 of those affected by Alzheimer's disease, multiple sclerosis, migraines, and depression worldwide are women? Women carry a heavier caregiving burden, both paid and unpaid, globally. Yes, we are women, and we differ from men in disease risks, frequency, severity, symptoms, diagnosis, and treatment response. Unfortunately, healthcare often overlooks these differences. Now is the time to revolutionize clinical development and digital health by considering sex and gender. This is the path to precision medicine and compassionate care. Imagine the heartache, the struggle, and the colossal costs to our healthcare systems. Picture the anguish of families and the loss of countless lives. But there is hope. Today, we commit to establishing a research institute as soon as possible. This is about more than research. It's about life itself. Our mission is to unravel the mysteries of sex and gender differences, advancing precision medicine and reducing suffering and hospitalizations. This is the mission of the Women's Brain Project, a global team of scientists, medical experts, and advocates. We are not just researchers, we're change makers. Leading us is Dr. Antonella Santuccione Chada, a beacon of hope with unwavering dedication. Her expertise in neuroscience, neurological disorders, and psychiatric diseases, along with her extensive healthcare leadership, including Roche and Swissmedic, drives our mission. We're here not just to conduct studies, but to change lives. We collaborate with industry partners, regulators, and governments seeking support. We share knowledge, host events, and raise awareness worldwide. Why the urgency? Because every day matters. Lives are at stake, healthcare costs soar, families struggle, and loved ones suffer. This is more than a mission. It's a lifeline. It's about answering the urgent call of life itself, demanding change, demanding action. Join us, and together we'll leave a legacy for a new world.
Okay, thank you, Laura, very much. Thank you both. That was amazing. And we can continue with questions. Please write your questions at the Q&A. And we can start with the first one for, I guess it's for Georgia. So the question is from Theophilus and asks, thanks so much for your presentation. I just think we may need to rest some of the findings using some other animal models apart from mice. I don't know if this has been considered.
Yeah, no, that's a great point. And in fact, there is a movement to bring in a bunch of different types of animal models. So one of the things that's been really important is to use non-human primates. And unfortunately, there's been a real decrease in ability to do that for a variety of reasons. But in addition, we've started to look at things like monogamous rodent species like California mice. We've looked at various vole populations, and even some researchers are working in pigs. So, you know, there's a variety of research into different animal models that really better maps onto things like the immune system of men and women, you know, lifestyle choices, parenting choices, all of which can end up influencing things like mood disorders.
Thank you. Emma, do you want to continue with the question?
Sure, I'd love to. Thank you. Thanks both, just echoing further thanks. So the next question is from Elvis, who wants to know if you're analysing estrogen effects in the brain, do you suggest to include male groups? And I think that's probably a question for yourself, Georgia.
Yes, absolutely. So males and females both have estrogen in the brain. And in fact, it's really been involved in, it can be synthesized within the brain. There's been beautiful work done by Catherine Woolley's lab showing this, and that this affects neuronal plasticity and the ability of neurons to fire. So it's definitely something that is influencing both sexes and should be looked at in both sexes.
I will just add something, if I may, Emma. I think Professor Holtz is more, I guess that we need sort of an educational plan for neuroscientists about how to formulate these essential questions. Because to my knowledge, I haven't had that course when I did my PhD, my master. So this is a really new, how can I say, multi-stakeholder, because it's not that we just need to add it from the biological perspective, because as you mentioned, now we need to incorporate more than two genders, more than a binary. What is happening with that population? What is happening with the population with hormone replacement therapies? And how can we, obviously animal models, but how can we model as well experiments using organoids, for example, using cell lines, for example, using microfluidic chambers. And it is actually, thanks to this webinar, thanks to all the audience that we are here, we are starting to think, okay, we have alternatives. So I think that it's important to start to stimulate this conversation. And I would like to hear your insight from this idea of a really educational plan about experimental design for neuroscientists, but also psychiatrists, etc., etc.
Yeah, I just wanted to add too that in trying to understand things like the mechanisms and how they may be similar or different, we're not just trying to improve women's health, right? When you look at something like the CRF inhibitors, that's something that could have been helping men for years who are having issues with mood disorders. And because they were tested in a group of men and women and didn't work in women, they're not on the market. And they really should be, a lot of these drugs should be reevaluated. Right? There may be things that we've already developed that could be helping people sex specifically.
Absolutely, absolutely, absolutely.
Thank you, Lara. I haven't had that course either. I'd be interested to know if any of our attendees have come across any similar courses. And I think often with this work, the role of men can often be overlooked. People are sometimes scared to say the wrong thing as well. So I'm getting lots of thanks as well in the chat as well. So just passing that on. But I want to invite you to just consider, as our panelists, maybe one thing that you might recommend. And I'll give you a bit of time to think about that in terms of thinking about that. And then I'm going to hand back over to Christina to take some more of the questions that we've had.
Yes, okay. And I agree. And I wanted to say again that we have also these open access videos that anybody can access and find a lot of useful information. So there is another question from Arman about Georgia. And the question is, when you were showing difference between male-female mice, were the males at 6 days chronic variable stress or carried out for the full male protocol?
So when we were seeing differences in terms of behavior, that was after 6 days of stress. So both males and females underwent 6 days of stress, and that's where we were seeing these effects in female behavior to a greater degree than males. When we go out to 28 days of stress, that's where we see behavioral effects that are the same between males and females. But that's also where we're seeing differences in things like transcriptional signatures, the behavior of neurons, and the behavior of non-neuronal cells, right? So together, those suggest, along with the human data, that the mechanisms that we need to address in terms of developing new treatments, both for males and for females, are different, and that we need to really consider this both when we're developing these treatments, but also when we're running our clinical trials, that we need to power our studies so that we can look within sex as well as between sexes or across sexes.
Yes, great, thank you. So the next one, I guess it's for both. And again, we have a lot of compliments, so thank you. And Saul asks if there are any tips to try to convince my colleagues and stakeholders to study SABV as primary questions and not just as post hoc sub-analysis. So we are working on it, but maybe you can also give your tips.
I can say in the States that there was really a push for many years that involved Congress as well as trying to get the NIH to mandate using female subjects in preclinical studies. That this took the work of many scientists lobbying for a very long time to get this instituted. And once it was instituted, there was a lot of anger about it in the scientific community, particularly because there was no increase in funding to now double the number of subjects that you had. But since it's gone into effect, a lot of researchers have discovered that things they've been studying for years are actually different in males and females. And as curious scientists, I think a lot of people have gotten excited about this. And so we've seen really this plethora of work coming out from excellent labs showing these sex differences and trying to understand what's causing them.
Lara, did you want to add at all? Or are you okay?
Yes, actually, I don't have an answer for that question. I think it's quite complex, but I think that how you can actually stimulate this conversation is, as I said, do not account for an innate variable as the biological sex might lead to an economic then noise data. So let's say that you have a hypothesis, you did not consider SAB, and then you have noisy data. And I am with my hat of researchers. Then with that noisy data, that means that I cannot do anything. That means that I might need to repeat the experiment. That means that I need more mice, more rats. That means that I need to do the experiment 3-4 more times to have a significant difference. You know, this is the kind of maybe questions that we can stimulate this conversation and also really brainstorm on the hypotheses. I think it's quite essential to actually sit with your stakeholders and have a literature review, because there are some papers that have been written since 1992 in the literature that have been showing actually that hormones have something to do in research in actually in schizophrenia, for example. So, and it has been hidden there, but it's not until that pieces of literature reach the people that is making the change, the economic decisions, that we see a change in policies, we see a change in funding. So translating these scientific insights to that population, I think, is what is going to drive the change forward. So good luck in that conversation.
Thank you. Ahmed has asked about the recording of this session, and just to let you all know that it will go on Alba's YouTube channel and website. And while you're there, you can look into Alba, you can join as well while you are having a look. So I'm going to hand over to Christina for, I think, probably our final question, so I'm just mindful of the time.
Yes, thank you very much. Now, we just had another one, but I don't know if we have time to answer both. So first question from Fedra is, I guess for Georgia, because about mice and rats. Can you elaborate more on convergent differences in male and female mice? What type of distinct mechanisms have been discovered?
Sure. So I think, you know, this
maybe I'm sorry, but the next one maybe is relevant because it's again about sex difference in rodents. But we'll see if we have time.
Okay. So the study that I mentioned, the 2015 study in Journal of Neuroscience in sequencing, as well as the 2019 or 2017 study in Nature Medicine by Labonte et al. Through the Nestler group that looked at this in humans with major depressive disorder. That's, I think, one of the first studies that really brought to head this idea of convergent sex differences, right? That the biology can be different between males and females. There's also been work, again, by Catherine Woolley, just looking at baseline signaling of GABAergic neurons and showing that that can be different in males and females. There's been a number of studies looking at how things like microglia shape the brain, specifically out of Peg McCarthy's lab and Stacy Bilbo's lab, where we see effects that are only occurring in one sex, even though the same behavior may be displayed in males and females. So for example, play behavior, which does occur in both males and females, seems to be driven by microglia and their eating of synapses in males, but not in females in the nucleus accumbens.
Thank
and
you.
I think, sorry.
Yeah, go for it.
I think also the work of Roberta Brinton about menopause is highly important, and Professor Mosconi, I think they publish amazing pieces regarding menopause and perimenopause, and for everyone here, you will be shocked. Very nice pieces of work.
Thank you. Our hour has flown by. So just to draw this to a close, just to thank our wonderful speakers again. Georgia, Laura, you were great. We will pop the recording up on the website, just to also thank my co-chair, Christina, and also all of the team at Alba and FENS as well for logistically making our webinar happen. And then finally, just to thank you, all of our participants, for joining as well. We hope it was an informative and interesting session. So thank you, everybody. And it's close enough to Christmas now that I can probably say have a wonderful Christmas. as well if you are celebrating. So thanks again for joining us, and we'll see you soon.
Thank you. Thank you all.